Hello everyone, today we will discuss about media preparation in microbiology. Media preparation. It is a cornerstone of microbiology, providing the controlled environments needed to grow and study microorganisms. It is the process of formulating, sterilizing, and dispensing nutrient-rich mixtures known as cultural media or growth media to support the life of bacteria, fungi, and other microbes in the lab. This procedure is essential because in a lab setting, microbes can't find the specific nutrients they need to survive as they would in their natural habitats. The prepared medium acts as a substitute, providing all the necessary components for growth. The essential components of cultural media. A well-prepared media is more than just a water. It's a careful balanced formulation containing all the fundamental building blocks for life. The essential components of cultural media are, first, nutrient source. This provides the main food for microbes. Carbon source like glucose or sucrose or for energy, while nitrogen sources like peptones or beef extract are for building proteins and nucleic acids. Another one is mineral and trace elements. These are vital for metabolic processes. Common examples include phosphorus for DNA and ATP, sulfur for amino acid, and a range of trace elements like iron, magnesium, and zinc. The third one is water. As the universal solvent, water dissolves all the other ingredients, making them accessible to other microorganisms. Let us talk about buffers. Most microbes are sensitive to pH changes. Buffers such as phosphate are included to maintain a stable pH as the microbes grow and excrete waste products, preventing the medium from becoming too acidic or alkaline. The last one is solidifying agent. This is optional. For solid medium, agar is added. This polysaccharide from red algae melts when heated and solidifies at room temperature, providing a firm surface for microbes to grow as a distinct colonies. This is crucial for isolating and counting specific type of bacteria. Now the steps which are involved in media preparation. The general procedure for preparing cultural media involves several critical steps to ensure the media is sterile and suitable for microbial growth. The first one is formulation. The specific ingredients and their quantities are measured out according to predetermined media. This can be done from individual components or more commonly by using a dehydrated commercial formulation. The second one is dissolving. The powdered or granular media is dissolved in distilled or deionized water, often with continuous stirring and heating to ensure all components are fully dispersed. After that is pH adjustment. The pH of the media is carefully adjusted to the optimal level for the microorganisms to be cultured. This is typically done by adding a small amount of acids, for example, HCl or base NaOH. Then moving on to dispensing, the liquid medium is poured into appropriate containers such as flasks, bottles or test tubes. Then moving to sterilization. This is a crucial step to eliminate any unwanted microorganisms that could contaminate the culture. The most common method is the autoclaving, which use high pressure steam at a specific temperature and pressure for a set period of time. Heat sensitive additives like certain vitamins or antibiotics are added after the initial sterilization using a technique like filter sterilization. Then moving to solidifying and storage. If a solid medium is being prepared, it is allowed to cool and solidify in a petri dish or test tube for slants or deeps. The prepared media is then properly labeled and stored under recommended conditions to maintain its integrity. Classification of cultural media. Media are classified in several ways which helps a microbiologist choose the right one for a specific task. The classification by physical state. It is divided into liquid media, solid media and semi-solid media. Liquid media is also known as broths, used for growing large quantities of microbes or for fermentation studies. Solid media contains agar and is used for isolating pure cultures and identifying microbes based on colony characteristics. Semi-solid media has a lower agar concentration, creating a soft jelly-like consistency used to test for bacterial motility. Now, the classification on the basis of chemical composition. The first one is chemically synthetic media, which is also known as defined media. Every single ingredient and its exact quantity is known. This is used for precise research on the nutritional requirements of the microbes. The second one is complex media, also known as undefined media. It contains ingredients like peptones or yeast extracts, whose exact chemical composition is unknown. This is common, rich media used for growing a wide range of microbes. Now the classification by function or application. The first one is selective media. Here it inhibits the growth of certain microbes while allowing others to flourish. For example, McConkie agar is selective for gram-negative bacteria. The another one is differential media. It contains ingredients that cause different microbes to grow with distinct appearances, such as changing the color of the medium or the colonies. McConkey agar is also differential, as lactose fermenting bacterium turn the colonies pink while non-fermenters remain colorless. The third one is enriched media. It contains extra nutrients like blood or serum to support the growth of fastidious organisms, those with complex nutritional needs that are difficult to grow. This was all about today's topic. Thanks for watching.